In this video, I'm going to give a basic introduction to the biotech industry. This is part two of a two-part series where I talk about the steps to actually get a bioproduct to market and the future innovations within the space. In the last video, I went over the three major biotech industries, the first being pharmaceuticals, the second being agriculture, and the third being industrial biotech. And so while each of these industries seem very different, they actually follow very similar steps to actually get a bioproduct to market. And so oftentimes you're going to start off with a discovery of some sort of bioproduct. Then that needs to be optimized in the lab to give you enough data and confidence to start getting a cell line to produce them at scale. These cells need to be further optimized within benchtop bioreactors that allow you to then go to scale manufacturing of a product, to then go ahead and start doing some pilot real world studies. And then once those work, then you can move on to commercial production and actually getting a product to market. And so the first step of this process is going to be the discovery step. And there are two ways to discover new biotech products. And so if you are a scientist with some sort of amazing biotech idea, one thing you can do is you can go find something in nature that's something similar and go ahead and extract that for your discovery step. This can be, some sort of some compound. This can be some sort of cell. This can be some sort of protein or enzyme. An example of this is if you have a system where you're trying to capture atmospheric CO2, then you can look at the enzymatic machinery of a plant and then try to extract enzymes from that and then use that as the first step in the bioproduct development process. Now, this is actually going to be relatively rare within the biotech industry. What's actually a lot more common is what you're going to do is you're going to generate some sort of random versions of those compound cells or proteins within the lab. You're going to determine which ones have the properties that you're looking for, and you're going to go ahead and isolate and optimize, later on the line to move towards commercialization. And so when you're talking about this process, you're going to oftentimes run into well plates. And so this is what a well plate looks like. They are like these little dishes with these little compartments where you can put little cells and compounds and aqueous solutions and reagents into. And the way you want to think about this is that each well is essentially a test tube. And so the way these things work is that you take some sort of random synthetic compound library or random protein library or some sort of random cell library. You're going to put each individual variation of those cells, compounds, and proteins into a well plate, and you're going to get a system that looks like this, where each well is going to have its own unique variation of that cell, protein, or compound. And what's going to happen is that you're going to have some sort of reaction that occurs with positive hits. And so the idea here is that if a well contains, for example, a cell with a certain therapeutic property, you're going to get some sort of color change or fluorescence change within that well that represents that, hey, this well actually has a cell that has the therapeutic properties that you're looking for. And so just for terminology purposes, this process is called a screen, and the reactions are oftentimes called assays. And the idea here is that once you go through the screen and once you have an assay that actually works properly, you go ahead and isolate the positive hits and then move on to the optimization step. And when you're talking about the discovery part of the bioproduct development process, throughput is super important. And the reason for this is that you can have oftentimes like even 100 million variations of cells, compounds, and proteins, and you're going to have very rare variations that are going to have the therapeutic properties that are going to actually produce the end therapeutic response within the patient, or have the agricultural properties that are actually going to make it to market. And so if you have a very low throughput, that means that you're going to have a very small search area. And so you're going to have something like this, where you're only going to look at a very small subsection of variations that doesn't necessarily overlap with the best possible variations that have the best therapeutic properties or agricultural properties. And so even if you were to repeat this process multiple times, the odds of getting a hit are incredibly low. And so more recently, what we've seen is the development of robotics that has enabled the increase in throughput by an order of 10x. And so previously, when you were talking about these manual processing of well plates, we were only realistically able to look at 100 to 1,000 different variations of cells, proteins, and compounds. And robotics was enabled us to look on the orders of tens of thousands to hundreds of thousands. And this started a field called high throughput screening, or HTS. And so if you want to kind of learn a little bit more about how this actually works and why the throughput is actually improved, I would actually go ahead and start looking HTS up online. And so Microtech has actually increased the throughput by another 10x. Examples of this are lab on chip technologies, and more recently, we've seen a lot of lab on product technologies. And so this allows us to actually look at, or on the orders of 1 to 10 million cells, proteins, or compounds that give us much more higher chances of finding a variation that has the best properties moving forward. So some of the challenges with the discovery step, the main one is the time and difficulty to develop the assay. And this is mainly because it's not super straightforward how you actually measure whether something has a certain therapeutic property or whether something is actually fixating nitrogen properly. And so if you talk to people, even in well plate industry, lab on chip industry, lab on particle industry, they'll always talk about how the time and difficulty to actually develop these actually assays or reaction conditions is actually the main bottleneck within this industry. But on top of this, a major problem is actually simulating real world environments and settings. And that's simply because a well plate is not the same as a human body or a farm. And so, finding ways to actually more accurately represent what happens in a well plate and what happens in the end use case is going to be an incredibly useful thing moving forward. And on top of this, the throughputs, even with some of these more, microtech technologies, is still not high enough. And so, like, oftentimes we'll have, infinite variations of cells, proteins, and compounds. And so, even though 1 to 10 million is a lot, we're still not, exploring the full diversity of these, different bioproducts. And so, any ways that we can increase throughputs even more than what is capable now is going to be incredibly useful in the discovery step. And so if you look back at the steps of bioproduct development, once you discover a bioproduct, it's usually not ready to start doing some real world studies. And so what you're going to do is you're going to start doing some additional lab optimization of those bioproducts. And so what again, these lab optimization does is it preps the product for real world pilot studies. And so what you're going to do is you're going to take that byproduct from your screening step. Oftentimes, you're going to do some rational editing. Maybe this is changing the structure of that molecule. Maybe this is changing the amino acid sequence. Maybe it's introducing a cargo into your antibody. Maybe it's introducing some sort of gene mutation within that cell. And then once you kind of do that, you're going to introduce that into a mouse, a plant, a cell, whatever system that you want to look at a lab scale. Oftentimes it's not going to work the first time. So oftentimes you need to go back and start doing some rational editing and then go back into the in vivo or in situ study and see, what that looks like. And then sometimes you even need to go back to the screening step. The idea here is that you want to repeat and iterate this process until you get enough data that convinces you that, hey, you are ready to then go ahead and start doing some more real world studies. Some of the challenges, one thing is that 90% of products actually fail here. And as I kind of mentioned, because it's so hard to, replicate, what happens in a well plate and what happens in a mouse, it's really unpredictable whether or not it's actually going to work. And so 90% of products actually can fail at this step. And it's mainly because, again, this process is very unpredictable and very nonlinear. So even if you're looking at two seemingly similar enzymes, they're going to require like completely unique processes to actually, get to the point where you have enough data and feel confident to move forward with. And so, again, this is again a kind of a brute force nonlinear process, and you're not going to know like how long it's actually going to take to actually get the data that you need to move forward. And the third thing is that there is also an extensive amount of labor and resources that go into this. And because, there's really no way of automating this step, and you just got to have, manually test everything, it's going to take a lot of labor, a lot of resources, a lot of time to actually move forward. And so looking back at the steps of bioproduct development, once you feel like you have enough data to start, moving to real world studies, the first step is you're going to develop some sort of cell or strain to produce that product at scale. And so just to keep in mind, so I'm only going to focus on the bioproducts that are going to involve the cell that produces or secretes that product or is the product themselves. I'm going to ignore a lot of the synthetic compound development, manufacturing processes. I'm also going to not talk about the personalized medicine field where you extract the cell from a patient and reintroduce it. I'm also not going to talk about the development of things like mRNA vaccines. And so there are six major producer cell types that you should be aware of. The first one is going to be bacteria. The species with regards to bacteria or E. coli and Streptomyces. There's also yeast, Cerevisiae and Pichia pastoris are the most common ones. There's also filamentous fungi, Aspergillus trichoderma. There's insects, SF9 and SF21 are the most popular ones. And there are also mammalian cell systems, so Chinese hamster ovary cells or CHO cells, or human embryonic kidney cells or HEC293 cells. And then there are also primary cells that you can use, like stem cells, immune cells are a perfect example of this. And so as you move up this table, you're going to have cells that have faster growth, higher production rate, better biological understanding, and less fragility. And so the general rule of thumb is that if you can produce your bioproduct within bacteria, you should use bacteria. And so why do then people use some of these more lower producing cell lines like mammalian cells? Well, as you move down this table, you're going to have cells that have much more complex machinery. And so the reason why this is important is, for example, if you are making antibody therapy, even though that that antibody can be made in a bacteria, there are certain post-translational machinery or modifications that need to be made to that protein, such that when you introduce it in a patient, it's not going to develop some sort of adverse immune response. And so, if you develop that and produce that protein in a mammalian cell, it'll work fine. Whereas if you're producing in something like a bacteria, it's not going to work fine. And so you need to find this balance between having a host cell produce enough of the product that you want to look at, but also have the particular machinery that's going to give you the properties that will allow you to get to market. And so the general rule of thumb here is that bacteria, yeast, and filamentous fungi are going to be used for agricultural and industrial biotech purposes. And then insect mammalian cells and primary cells are going to be mostly used for pharmaceutical purposes. And so again, there's going to be some exceptions to this. So for example, there's a lot of therapeutic proteins that are produced in yeast and bacteria, but this is going to be the general rule of thumb. And so some of the specific applications, so bacteria are often kind of going to be used for a lot of the pathway non-pharma proteins applications. Yeasts are going to be used for pathway non-pharma proteins as well. Cerevisiae in particular are used for a lot of pathway applications. Pichia is oftentimes used for a lot of proteins, and in particular biopesticides and a lot of alternative proteins. Filamentous fungi are really popular for a lot of non-pharma proteins like enzymes. There's also things like insects that are very popular for pharma proteins and bioproduction for gene therapies. CHO cells are really popular for antibody proteins, and HEC cells are really popular also for viruses for gene therapies. And stem cells and immune cells are mainly going to be limited to cell therapy applications just because they're so fragile. And so, finding cells in more mature industries is a little more streamlined. And so you're going to have these commercially available host cells, oftentimes that you can just directly buy that has all the machinery for high production, proper folding, et cetera. And then what you can simply do is that once you buy those cells, you can transform or transfect them with the proper genetic machinery to produce the product that you want to look at. And then you can run through a simple screen in order to find out which ones produce the product at the highest production rates. But if you are looking at a new product, and so for example, a lot of these pathway engineering applications, it's going to be a little more complicated, and so you're going to have to go through kind of this iterative process of screening, rational editing, and testing in order to make sure that those cells produce the product at a high enough amount and with enough purity to actually move forward into some of these more large scale applications and experiments. And so one thing that you should also keep in mind is that as you're talking to people in the field, cell line development refers to this step in regards to mammalian cells, whereas strain engineering refers to the step in the context of microbes. And so some of the challenges here, one is that 20% of products actually fail to find a host producer cell, and this is because you have certain products that are actually toxic to cells, and so it's going to be incredibly hard to find one that's actually resistant to the toxicity of that product. Maybe the product has a certain folds that are not capable of being produced within its producer cell, and so there's actually a lot of products that actually fail simply just because they cannot never find a host producer cell to make that product. Another thing is that there's a lack of single cell colony screening tools that select based on production. And so a lot of these screening tools are mainly focused on the discovery of products and not actually on the production of that product. And so that's a huge challenge within the space right now. Another thing is if you talk to a lot of these cell line developers and strain engineers, is that there's a lack of genetic tooling and biological understanding to do more rational design or direct CRISPR editing of cells. And so we need a lot more genetic tooling and understanding here to actually use this to get to the levels that we need to. So once you have a producer cell type, then you want to go ahead and start optimizing your benchtop bioreactor conditions that allow you to get to pilot scale manufacturing. And so, as we kind of think about how, a bioproduct is actually made in the biotech industry, they are essentially made in what's called a bioreactor fermenter. And these are these large tanks. Bioreactors refer to mammalian cell systems, fermenters refer to microbe systems. And the idea is that these tanks have precise control over something like temperature, pH, nutrient concentration, and a bunch of other parameters. And the general concept is that you want to grow up these host cells in these large tanks, have them, produce that product. And the idea here is, again, cell culture refers to mammalian cell systems, fermentation refers to microbe systems. But once you have enough product to go ahead and move forward with, you then extract and then do your pilot studies, your clinical trials, your farm studies, whatever. And so this is kind of what these tanks look like in the commercial setting. And so again, these huge tanks that can cost, $100,000 per production batch. And so before you ever get to this type of scale, we really need to think about optimizing the bioreactor conditions and media composition and strain performance first, mainly because, you want to make sure that you're producing the most amount of product when you start getting to the scale and making sure that product is pure and a bunch of other things. And so the universal first step here is going to be the Amber 250 system. And so these are these automated bioreactor systems. When the basic idea here is that you have a bunch of mini bioreactors that are on the scale of 100 to 250 milliliters that have a lot of these controls that you would see at the larger scale, but again, scale down so you can actually start running smaller studies. Amber 250s have these automated arms such that you can, introduce reagents at a certain time, take samples, things along those lines. And the basic concept here is that you can, try different cell variations and figure out which ones, produce the most within these mini bioreactors. You can try different media compositions, different bioreactor conditions. Maybe you introduce feeds at different times. Maybe have different formats of bioreactors, things along those lines, to figure out which ones actually produce the most. And so again, the goal is that you want to find conditions that achieve economically viable quantities. And what you're going to do is you're going to use something called technical economic analysis or TEA that projects the economic viability of products based on what you're seeing at the small scale. And so, example of this is you can have a plot of time of production and amount of product being made over time. And then you're going to have some sort of target that you need to reach in order to be economically viable. And so you can run your first Amber 250 bioreactor test and then get a plot like looks like this. Oftentimes you're not going to get to the point where it's economically viable. And so you're going to, change the media composition. Maybe it's the cell line they're using, and then hopefully over time you can actually get a certain combination of conditions, and bioreactor machinery and cell variation that will give you the titers that are actually economically viable when you start projecting when you start going to pilot scale. And so the challenge here is actually going to be from going from 250 milliliters to 1,000 liters. Again, so if you can kind of compare scales, there's going to be different issues that happen at the large scale that are not represented in the small scale. For example, you're going to have a huge pressure difference at the top of larger bioreactors compared to the bottom. And that's mainly because you're going to have so much weight from the media in these larger volumes. And you can actually have situations where the cells actually start dying at the bottom of the tank because the pressure is so high. Another huge thing is that it's also very hard to keep nutrients and gas uniform at these larger scales compared to a compared to a smaller setting. And so oftentimes these cells are being exposed to different conditions over time when you start comparing to something like an Amber 250 bioreactor. And so what a lot of these bioprocess and fermentation engineers need to do is that, they need to, start going from, okay, 250 milliliters, then go to 1 liter, start optimizing there, then move to something like maybe like a 2 liter or 10 liter, and then optimize there, and then slowly move their way up to 1,000 liter pilot scale reaction. And so, again, every company is going to have their own very unique process and philosophy behind this. But the general idea is that over time, you're going to slowly move up to the point where you're confident to start running these larger pilot scale studies. So some of the challenges here, one is that even with the Amber 250, we simply do not test enough conditions. Right now, we test on the order of 10 to 50 different conditions, where we need to get to the scale of 100 to 500 different conditions to be really confident that we're using the best possible conditions to produce the most amount of product when we start doing pilot scale. On top of this, because each product is going to have its unique challenges, there's extensive amount of labor, time, and resources, or to get to the point where we're confident to start running pilot scale studies. Each experiment is going to cost $10,000 plus, and so this makes it really, really hard for us to run many experiments to make sure that we're again getting the best possible results at the end of the day. And then another major problem is that you can't actually measure the production of the product in real time. And so the idea here is that you put your cells into a bioreactor, grow them up, have them produce the product. But the problem is that you don't know how much product is going to be produced until you actually measure it afterwards. You can't actually measure how much product is being produced while it's being produced within the bioreactor itself. And so we need things like biosensors to help us, optimize these conditions. Looking back at the steps, the next step is going to be pilot scale manufacturing. And so, the first part is going to be what's called upstream processing. And the first step of upstream processing is getting enough biomass. And so what you're going to do is you're going to go to your cell bank, get a frozen vial of cells, grow them up in a small vessel, move to a slightly larger vessel, grow those up, moving them up to even larger vessel, grow these up, and then finally get to your end bioreactor fermenter tank, and then grow them up to a density that you're looking for. Just for reference, this is what's called something called a seed train. So once you get to this step, you're going to go ahead and induce production. And so as you're growing up the cells through the seed train, you oftentimes those cells are not producing the product that you're looking at, because you want the cells to be purely focused on growing to the point where they can actually get to density within these larger tanks. And so what you can do at this step is add some sort of inducer, and this is going to be some sort of chemical or reagent that actually causes the cell to start producing this protein. And so, once we've, produced enough product within these tanks, we then move on to what's called downstream processing. And the step one is downstream processing is actually the separation of the cells from the product that you want to look at. And so oftentimes what this is done is that you're going to move over to the centrifuge, and this will allow you to, again, pellet down the cells and separate it from the byproduct. And so just, for reference, this is kind of what they look like in the scale that we're talking about. You can actually even have even larger centrifuges than this. And so, in the previous cases, I've kind of shown what happens when a cell is being secreted into the environment, but what happens if a byproduct is actually being accumulated within the cell? How do you actually isolate that? Because if you just simply centrifuge them out, the byproduct is going to be pulled with the cells too. And so what you need to do here is you actually need to find a way of like lysing or rupturing the cells to release the product. And so the preferred method here is going to be chemical lysis. But you might actually have situations where the chemicals that you actually introduce to the cells to rupture the cell membrane might actually damage the product itself. And so if this is the case, what you can do is you can use some sort of mechanical lysis. This can be something like a homogenizer, where you can actually like physically actually rupture the cells. And it's like, again, one way of doing it. And so again, if you can use chemical lysis, that's going to be preferred method because it's just a lot cleaner than mechanical lysis. But again, but you're going to be forced into a situation where you're going to have to use mechanical lysis as well. And so then the second step of downstream processing is actually the isolation or purification of that product. And so, I kind of show, I guess, separation of the product from the cells, but this broth is not going to be pure. And so there's going to be like other molecules and proteins in the broth that you actually need to get rid of before you actually go ahead and start doing your pilot studies. And so one way of purifying your product is that you can have what's called a column and run through a column chromatography experiment, where you have a tube that has beads that specifically bind to the product, and such that when you run the broth through this tube, the product that you're looking for remains in the tube because they're bound to the beads that are represented here, whereas everything else actually flows through and causing a separation between the product and everything else. And then later on, you can then isolate the product and then move forward from there. And so again, this is kind of the scale and the scale and what they look like in the industrial setting. And so, one thing to keep in mind is that columns are limited to pharmaceutical applications, and that's mainly because these are simply too expensive for agricultural and industrial biotech. And so, in the pharma world, you can make a small amount of product and charge a lot, whereas in a lot of like agricultural and industrial biotech settings, you are have to make a lot more for cheaper. And so, even though these are like really really effective at purifying the product, they are simply too expensive for the agricultural and industrial biotech space. And so oftentimes what you can do with these other applications is you can add salts that cause precipitation of the product that you can then isolate down the line. Another way of doing this is you can do what's called tangential flow filtration, or TFF. And the idea here is that you have some sort of tube and then you have some sort of membrane that you can run all your broth through. And so the product that you're looking for can pass through the membrane when everything else can flow through as well. And so this is going to be a lot less efficient, and you're going to get a lot less purity than something like a column chromatography system, but it's also going to be a lot cheaper. And so just to kind of visualize this is kind of what a TFF system looks like in real life. So some of the challenges here, one is that 20% of product can be lost in each purification step. And so if you needed, for example, to have multiple columns to actually isolate the product that you're looking at, you need to find this balance of purity and losing product. And this is going to be, again, a huge problem, because when you start getting to commercial scale, you're talking about batches that are worth $1 to $10 million, and so 20% is a huge cut into the revenue of the company. Another thing here is that each pilot batch production costs on the orders of $100,000. So that's a major challenge. And that's why it's also very important to optimize all the conditions upstream, and so that when you get to this step, that you're using this costs and these prices effectively. Another thing is that seed trains can take a long time, particularly for mammalian cells. And it's just because mammalian cells take doubling times of like 24 hours, and so you might actually have to wait up to a month or even 2 months to have the cells grow enough to actually start getting to the production step. And this is going to reduce the amount of batches they can do per year. And another major problem that's a little bit less technical is actually there's a huge lack of training and education in academia. And so academia really likes to focus on the discovery and lab optimization step. That's because, again, that's the research and the discovery of these various bioproducts. But when you're talking about the biotech industry, most of the effort is actually put into the manufacturing of bioproducts. And so we really need to think about how do we actually educate people and have people learn about how the manufacturing process actually works. Because if we don't, we're going to actually have a huge delta in the workforce in terms of how much labor we actually need and what the workforce is actually available. And so we really need to start educating people in academia about these, bioprocessing and fermentation steps. And so, if we look at this, once you go through the pilot scale manufacturing, you're going to have enough product to start then doing some more pilot scale real world studies. And the idea here is that you're going to test the products in the real world. And so if you're talking about a pharmaceutical product, you're going to run your clinical trials. If it's an agricultural product, you can run your field studies. If it's some sort of food product, then you're going to start running taste studies. Again, this is going to be very unique to the particular application that you're looking at. But the major challenge here is, again, like 90% of products fail at this step. And so this is a major challenge is because you can go through, like, again, all these steps to engineer cells, optimize your bioreactor conditions, spend millions of dollars, take years, and at the end of the day, your product might not even work. It might not have the therapeutic response that you want. It might not produce the results in the field that you want. It might not have the taste that you want. And so this is a really big problem within the biotech space. And again, it's also very unpredictable. Like you have no idea predicting how the end user is going to take it, or like the result that you're going to get. And so, finding ways to make this more predictable and reduce the failure rate is going to be a huge thing that we need to figure out in the biotech industry. And so, once you run these pilot real world studies, and you're lucky enough to have something that actually has commercial viability, then you move on to commercial production. And so this is when you start building again these like 10,000 liter reactor facilities. They can go up to like even like a million reactors, a million liter reactors. And then you also start developing distribution channels to actually distribute the product throughout the world. So some of the challenges here is the facilities cost $100 million to make, and so that's actually like massively reduce the number of products that you can actually move forward to market. And so you need to make sure that again these products are actually economically viable before you move on to the step. These facilities can take 2 years to build. If you are working in the pharmaceutical space, you're going to have heavy regulation, so you need to make sure you have good manufacturing practices and other components as well, and also maintaining sterility. And so, if you actually talk to a lot of people who work in the manufacturing space, their actual biggest concern is maintaining sterility, and that's mainly because each batch is worth $10 million, and so if you can contaminate, you're going to ruin that entire batch, and that's going to be really upsetting for the company and be a huge cost driver. And so that's again the overview of all the steps of how do you get a product to market. And so one thing that I think kind of want to highlight here is that startups oftentimes don't have expensive facilities. And so oftentimes you can have like an academic scientist with a great idea, but they don't have the expensive screening facilities or expertise, or the bioproduction facilities to actually produce the product and actually get to market. And so what these people can do is they can partner with contract organizations who will have all this infrastructure and expertise in place to help them get to market. And so, just for terminology's sakes, if we're talking about a partnership in the discovery or lab optimization step, it's called what's a contract research organization, or CRO. If you are working on the more manufacturing side of things, usually you go to a contract development and manufacturing organization, or CDMO. If you're getting help on the pilot scale studies, like a clinical trial, they're also called CROs. And then when you're talking about these, 10,000 liter plus manufacturing applications, you're going to oftentimes partner with CMOs. And so I talked to a bunch of people in biotech, and what I really found surprising was that the biggest problem is costs. And so I did like 287 interviews and actually quantified what each one said was the biggest problem. And they all said that essentially cost manufacturing development is the big issue. And this was if you talk to antibody people, gene therapy people, pathway people, agricultural people, they all said that cost manufacturing are the really big problem within the industry. And why is this? Well, we can make all these gene therapies, for example, that can cure sickle disease, but they cost like $2.2 million. We can make a 1 pound culture meat burger that amazing and tastes good, but it costs $100. And so even in biotech, we can make these incredible products that are incredibly useful. But at the end of the day, if they are too expensive for an end consumer to actually buy, it's never going to be adopted. And so we need to really find ways to reduce the costs. And so where do these costs come from when we look at all these steps? Well, when you're talking about the discovery and lab optimization and production strain development steps, it's largely going to be driven by the large failure rates. So you can imagine it's going to take, roughly like 10 times or 10 times the experiments to finally get a product that actually has economic viability or commercial viability. Another thing too is that when you start talking about optimizing benchtop bioreactor conditions and doing pilot scale manufacturing. This is largely driven by like labor costs because again, like each product is going to have its unique challenges, and a lot of it's not automated quite yet, and so there's a lot of costs that go into labor in order to do the R&D for these things. We're talking about real world studies. Again, you're going to have like really high failure rates, and so after you go through discovery and bioprocess optimization, you also are like most likely to fail at that step, and so that's going to drive costs up a lot. When we're talking about commercial production, as I kind of mentioned earlier, it's like $100 million to build a facility, and even if you go through all the steps, you can even get to a situation where the amount of product that's being produced is simply not high enough to be economically viable. And so how do we actually solve this? And so knowing that, the costs are the biggest problem within the biotech industry, we can anticipate where the future innovations are actually going to come from. And so my opinion is that this is going to come from the cell engineering and equipment and also machine learning. And each of these are going to find ways to reduce the costs to make these products. And so how is cell engineering going to reduce costs? So one thing to keep in mind is that cells are not designed for this purpose. And so a mammalian cell or animal cell is meant to grow in some sort of body or animal. A microbe is supposed to be growing in the ground. These cells are not supposed to be growing in tanks. They're not designed to grow in tanks. And now the only exception to this right now are CHO cells for antibody production. And the only reason why this is that it's because we spent 30, 40 years to actually engineer these cells for this particular purpose. And cells for other applications simply just have not had this much engineering or had this much history to actually make them, useful for this particular purpose. And so, one approach that people are also looking at are cell-free systems. And so the problem when you're looking at producing product within a live cell is that you're limited by what maintains cell viability. And so, certain conditions that result in high production may actually, kill the cell. And another problem is that, certain metabolites might not enter the cell or might be inefficiently enter the cell. And so one thing you can do is you can, instead of taking live cells, you can simply lyse the cells, and where the lysate has all the enzymes, ribosomes, et cetera, for production, and you can fill up these bioreactors and fermenters with these lysates. And so you're going to have all the components that are necessary to produce the product while not being limited by what maintains the cell viability. And so what are the challenges with cell engineering? One is that, when we took looking at traditional cell systems, there simply just, again, has not been enough time to make sufficient host cells for these applications. There's also a lack of genetic and screening tools. And so it makes, the time it takes to actually engineer new cells incredibly long. And when we're talking about cell-free systems, they don't have, because they're, dead, they don't have the ability to replenish enzymes and cofactors that a live cell would naturally do. And so if you want to produce for longer periods of time, that's going to be a huge challenge. And another thing that's a problem with cell-free systems is that you're limited to more simple pathways and expression systems. And so it's going to be quite a while before we get to a point where we can use cell-free systems to start producing something like pharmaceutical proteins. And so, another area that we're going to see a lot of innovation in is the equipment side of things. And the reason for this is that the current equipment is simply not designed for certain areas within biotech. And so we have GMP grade equipment for pharmaceuticals. We have a fermentation equipment for beer production, but we don't really have equipment that is specific for agriculture and industrial biotech. And so, for example, the GMP, your highly regulated pharmaceutical equipment is probably too expensive and is overregulated relative to what we actually need to produce, like for example, like a cultured meat burger. Whereas like on the other side of the spectrum, like a lot of fermentation equipment might not have certain tooling or regulatory components to it that make it suitable for a lot of these other applications. And so we really need to design new equipment for these particular applications. Another major problem is that we currently use what's called batch production, and which is very inefficient. And so again, the basic idea is that you grow up these cells into these larger tanks, and then have them produce, and then extract from there. And if you want to make more products, you actually have to repeat this entire process over and over again. And so you can imagine, like when you're talking about pharmaceutical proteins and you're using mammalian cells to grow up and they take, months to grow up into these large tanks, each batch is going to take several months to make, and so you need to find a way to more continuously make these products. And so again, we need to move on to more continuous systems. And so instead of having a closed off tank where this products are being made, we can have these bioreactor systems. They have some sort of external compartment that actually extracts some sort of small amount of cells and product that can then be sent down to downstream processing. Any dilution in these continuous systems can be replenished by simple cell growth and continued production of the bioproduct, such that you can again take a small sample down the line and then continue this process for longer periods of time. And so the basic idea here is that you don't have to wait each time for the cells to grow up into these larger tanks. You only have to wait once, and then you can run these systems for longer periods of time. So some of the challenges here is one, it's really hard to maintain sterility for a long period of time. But the more important thing is that oftentimes the recombinant proteins, pathways, and other machinery that we actually introduce to the cells is actually toxic. And so what ends up happening is that the cells start to evolve to stop producing the product within a week. And so even if we can have a continuous reactor system that can continuously extract products for example, like several months, the cells will actually stop producing within a week. And so again, we need to get to continuous systems that run for like 1 to 6 months in order to be economically viable. And so we need to see either innovation in engineering cells that are more compatible with these introduced machinery, or we need to see innovation on the equipment side that allow us to continuously produce product for long periods of time. And so I think that again, the main and major source of cost reduction is actually going to be the advent of machine learning within the biotech industry. And so the reality right now is that wet lab is very challenging. Biology is very sensitive. It's very hard to make biology repeatable. And right now we really kind of use brute force and kind of hope we get lucky. And the reason for this is that if you look at a cell, cells are simply too complicated for us to understand. There's simply no way for a human to know exactly what to do in order to get the cell to produce enough product or have certain properties that make it economically viable. And another thing too is that if we look at a cell in a bioreactor, the cell behavior here depends on the genetics, it depends on the media composition, depends on the reactor conditions, depends on the history of cell, and a lot more properties. And the current setup right now only allows us to test each of these effects individually. And so, for example, a well plate only really tests for genetic variation, and an Amber 250 only allows us to test reactor or media compositions. But all these variables are dependent, right? And so maybe if like the best genetics in a well placed setting is not the best genetics in a certain media composition as you scale up. And so we really need to find ways to actually start looking at all these dependent variables simultaneously. And so what I actually believe is that machine learning will end up replacing a lot of these lab processes. And so just to kind of give a brief overview and high level idea of how machine learning works. And so let's say we have some sort of real world data, and let's say it's how titers are dependent on temperature. You can form some sort of equation that allows you to model it or represent this data. And so then if you have a third component, you can have more data. You can have update the equation such that you can get a much more complicated model that represents the data set. And so the basic idea here is that you have some sort of equation that allows us to predict the results in areas that we don't have data points for or have not actually collected run experiments for. And the more high quality data that there is, the more accurate the equation. And so what a neural network is essentially a more advanced version of this concept. And so instead of just relying on mathematical equations, you have a combination of mathematical equations, algorithms, and computational tools that allow you to model what's happening in the real world and make predictions in the real world based on the data sets that you give it. And so again, computation models describe the input data. You can look at a lot more dependent variables. You can start looking at more nonlinear relationships. And these models can actually change dynamically as you introduce more or new data. And so the basic idea is that we can take all the data that we're getting from these, these screening processes, whether it's like, a robotic screening facility or an Amber 250, and we can just use all this data to start developing computational models that allows us to predict results based on the desired inputs. And so again, if we compare the wet lab process to a machine learning process, the method of testing in a wet lab is going to be physical, whereas machine learning, you're going to use computational methods. The type of variables that you can explore in a wet lab are going to be independent variables, whereas machine learning can look at dependent variables. And the number or tests or variations of cells, proteins, or compounds that you look at are on the orders of hundreds of millions, hundreds to millions within a wet lab setting. But in machine learning, you can actually look at trillions of different variations. And so how is this going to reduce costs? Well, if you switch from physical systems to computational systems, you reduce the amount of labor, materials, and time necessary to actually develop these products. And so that's going to significantly reduce costs. If you're looking at, dependent variables instead of independent variables, you're going to decrease the failure rate, which is also going to reduce costs. Also, if you're looking at trillions of interactions versus just simply like hundreds to millions, you essentially ensure that you're getting the highest possible titers and best therapeutic effects. And so this is also going to reduce costs over time as well. And so some of the challenges here, one is that we have a lack of computational power to actually do something like this. So right now we're kind of limited to like a little more simple systems. Again, we take like internet data and use computational, develop like large language models in order to, develop these algorithms that can make interpretations. And so when we're talking about biological systems, they're a little more complicated, and we simply don't have enough computational power at the moment. Another problem is that collecting enough data is really hard and also takes time. And so there's no readily available database that everyone can tap into. And in the biotech space, developing the assays and developing the experiments to actually collect the data is really hard. And so the speed at which we can actually get enough data to actually make really powerful models is going to be really challenging. And I think the third thing that is something that people are not considering is that it's going to be really hard to monetize these ML and AI tools. And so ML and AI are very deflationary. And so it's going to be hard to have companies that actually make ML and AI tools that are readily available to everyone that has a model that's actually economically viable. And because that's going to be really challenging, it's also going to be hard to get investment, I think, over the next few years. And so finding ways to actually monetize machine learning tools is going to be incredibly important moving forward. And so that's going to, again, wrap up the future innovations. Again, this is going to wrap up the talk altogether as well. So comment below if you think I missed anything. If you missed part one, make sure you go to the Coral Lab UCLA on YouTube. And I'm also going to be posting this up on my LinkedIn page as well. So thank you very much.